Here we are, Chapter 10, Part C. We are going to move into the special senses, and it will be a two-part as well. So we've got a 10A, a 10B, a 10C, and a 10D. One thing I would like to emphasize, part of the reason why we spend so much time on special senses and so forth is due to the fact that they are so much a part of our existence. So when you're treating patients or you're dealing with people that you care about and so forth, part of our quality of life is how we perceive it. So, for example, we're moving into the winter season here, and this is in Quebec City, near Montreal. They actually pave over the normal walking paths with water to create these ice paths in the city park there. So it's absolutely beautiful. And I think a lot of our quality of life is certainly our how we approach life, our attitude, our state of being in terms of our mood, but also in terms of how we experience things. So, in your interactions with patients and clients and those whom yourselves and those whom you care about, attention to how we perceive is, I think, an important adjunct in and above whatever it is you might be doing to help with their deficit or their problems. One thing that makes the special senses different from the somatic senses is that you have dedicated sensory cells rather than free nerve endings. That said, there are two general signaling pathways that signals will be propagated in these cells. For the auditory and vestibular pathway, we are looking at ionotropic types of responses with hair cells. So when we talk about hair cells, we're talking about specialized cells that have mechanoreceptors. This is a figure from chapter 6 where we had a stimulus, and it looks like a chemical here, and that's what it was originally. But in this case, I've replaced the text with stimulus, and it's a mechanical stimulus that then causes depolarization. For most of the other special senses, if not all of the other special senses, you're dealing with metabotropic situations. So for vision, smell, and taste, we're talking about metabotropic processes that cause depolarization and then signal propagation. So that can include G proteins or the creation of intracellular molecules. It doesn't have to be G protein mediated, but most are. And then that will then lead to into a change in membrane polarity, a receptor potential, if you will. So things like cyclic AMP, calcium, ATP, those are all, and you'll see these. I want you to take a look at the figures, the mechanisms as we go forward. We're only going to drill down into primarily vision, because that's the one that sort of dominates in our society. And so we're going to deal with that mechanism. But you will see these second messengers in the other mechanisms that I show you. In any case, what happens is we get a membrane potential that leads to some kind of an electrical signal or something along those lines, leading to exocytosis of neurotransmitter. So we can have ionotropic and metabotropic responses, and I just want you to remind you of that, and hopefully you'll key into those in these slides. What we're going to do now is a survey of each of the special senses, talking about the key features, what makes them what they are, talking about maybe some of the mechanism or some of the things that are involved those senses. Just recently, we have seen that people who, not everyone, but many people who are exposed to an acute infection of the coronavirus, COVID-19, or also more correctly, the SARS-CoV-2, they lose their sense of smell and or taste. And we weren't entirely sure why that was, but it seems to be related to inflammation, so that there's some kind of an inflammatory process going on that is then leading to a, shall we say, a sequestration of stimulus to the receptor. On the other hand, I have known people who have had nasal injuries who said that the world just exploded in smell during the time that immediately or surrounding the time that they had, like for instance, got hit in the nose with a soccer ball, that sort of a thing. So it's hard to know exactly what's going on, but an interesting phenomenon that's occurring here in real time. So let's segue into what we consider to be the oldest special sense, that of olfaction. The reason we consider it the oldest is that we can follow the evolutionary tree and we will see the sense of smell in the most primitive and the oldest of the multicellular organisms. And that may be also why there's a stronger connection between the sense of smell, memory, and emotion. There seems to be a connection with that limbic system, the hippocampus, the amygdala, the dentate gyrus, the what we call the lower order neurons in the brain itself. And that makes a lot of sense if the olfactory system is the oldest. Um, just for your own recollection, you can probably recall a smell that reminds you of your grandparents' house or a place that you like to visit. Like for instance, if I go out west into into Montana, there's the smell of the sweetgrass out there that just will just knock me over every time I smell it because I really enjoy being out there. You can get those sorts of things, nostalgia and so forth. The interesting thing about olfaction, which makes it a little bit different than the other special senses, is that these are actually sensory neurons. The cells that are the special cells are neuronal in nature, and they are supported by an olfactory epithelium. The great hope when it comes to neuronal regeneration arises from the olfactory neurons because these are neurons that actually retain the ability to regenerate themselves. The odorants that we smell, and we can smell on the order of trillions of different smells. We can only taste about 5, maybe there's argument for a 6th flavor when it comes to gustatory perception, but the minimum number of smells in a normal person is somewhere on the order of 10 to 12 trillion different smells. Partly because we have G protein coupled receptors that are doing this, and we can alter the 7 transmembrane domain and get responses there. So it's a really astonishing thing. Super tasters, these folks that have extraordinary tasting and ability, like wine connoisseurs and that sort of a thing, they can maybe sense up to 20 or more trillion different smells. But it's metabotropic. So GPCRs, metabotropic, that's kind of the motif, pretty much all the senses except your ear and balance, which are basically in the same area, are metabotropic. And so here's just a figure from your book showing you that these olfactory neurons project into an epithelial support area. That's also one of the reasons why you really can't discriminate location with smell very well. Even dogs that have extraordinary senses of smell, they don't really locate certainly by population coding like we do with our hearing. They do it by intensity. So there is maybe some population coding going on, but it's intensity related rather than timing related like in auditory senses. And so those neurons will project onto the olfactory epithelium, and that's where, and there's a layer of mucus here, which is why oil, lipophilic molecules have much stronger scents by and large than aqueous solutions. Water, if you have really pure water and it's in a nice container that doesn't have any plastic or anything in it, doesn't have any smell at all because it can't penetrate that somewhat lipophilic mucus. But rose oil or olive oil, or like if you're grilling a hamburger or something, you get that odor, or fish. You get the house or onions are all very lipophilic. So that's why oil-based odorants tend to be much more potent than water-based. But it's convergence here. It's another reason why we can't really locate from population coding other than by intensity. The point of this figure is to show you that the receptors, the chemoreceptors, are embedded in, are encased in this mucus layer, and the odorant molecules must be able to diffuse into that layer. If they can't diffuse or they don't diffuse very well, then they will not be perceived. When it comes to taste, it turns out that we have 5 major tastes, but these are also metabotropic. They are GPCRs, like mentioned in the olfactory system. What's interesting about gustation that is a little bit different than the other ones is that a lot of the other ones are believed to be neuronal in nature. In the case of gustation, it is actually non-neural epithelium that are our taste receptors. So a little bit of a deviation here. And I think that these little factoids help us to kind of discriminate between the different senses. Turns out we only have 5 major flavors, whereas we have literally trillions of different smells. And that just blows me away. One more little thing I'd like to emphasize here with umami. Umami is a sort of peppery, savory flavor that has a lot of notoriety because for decades now there's been an urban myth about sodium sodium glutamate. That is to say MSG, monosodium glutamate, which is actually patently false because glutamate is in fact an amino acid. We use it all the time. It's involved in neurotransmitters. So this, is it possible there's an allergy? Yeah, unlikely. Doesn't mean there isn't. I've seen people put water on their skin and develop a rash. I mean, what are you going to say to that? But in any case, the things to remember about taste are the different types of tastes. They are non-neural. They're metabotropic, just like smell, just like vision. You can develop specific appetites to these things. These things tend to drive complex behaviors and psychologies, cravings, those sorts of things.
Used to be that they thought that different types of taste buds were found on different parts of the tongue, but that doesn't seem to be as true when looking at the cells at a deeper level. What we can see is that there are different kinds of taste receptors in individual taste buds. And so let's take a look at that a little bit closer. There are 5 basic flavors that can be found in any given taste bud. These include the salt, presence of ionic compounds, particularly sodium, sour, which is the presence of acid, bitter, which is usually a base or an alkaloid of some kind, sweet, which is the presence of simple and short level carbohydrates, sugars, and then this fun one, which is a Japanese word, umami, for deliciousness, which is a glutamate receptor. So it detects a type of amino acid and gives things a savoriness, like the difference between, say, just straight up ham and pepperoni. That sort of peppery, savory flavor is what umami is turned out to be, and that's one of the reasons why we find a lot of glutamate flavoring in Asian cooking. There is an urban legend about sodium glutamate or monosodium glutamate, MSG, and it is in fact an urban legend. There have been many, many rigorous studies showing that monosodium glutamate does not cause allergies, but yet for 50 years or more, the urban legend has persisted.
Just to give you a perspective on what makes this so interesting, probably the world's first millionaires were these folks that were creating and importing what we call fish sauce. That's this one right here. These fish stocks are actually high in glutamate. They are high in sodium glutamate. They are contributing to the umami, that flavor. They are flavor enhancers. And so, like I said, the world's first millionaires were probably these Romans who were catching fish and then fermenting them in these jars 3,000 years ago.
And you don't have to learn the mechanism here, but the basic story is that we have metabotropic signal transduction. We're getting some type of a signal that's leading to a second message, leading to exocytosis, GPCR, second message, ATP synthesis, or exocytosis. Next, we're going to move into the mechanoreceptor side of the equation with hearing and balance. Before we get started on the mechanism per se, just understand that what's happening, especially in hearing, but to a lesser degree in balance, is that we are transducing physical waves of pressure and less pressure from air into a liquid wave that then causes hair cells to move and open and close. Sound waves are alternating pressure and what we call rarefaction. So we have compression, where you see molecules stacking up, and then rarefaction, where there are areas of slightly less pressure. And if we look at a tuning fork and how it moves, when it moves closer together, they create pressure waves and so forth. And that's what sound fundamentally is. It's a combination of compression and rarefaction in air most of the time, sometimes in water if we're underwater. So there are two aspects of sound if we're talking about qualitatively what sound is. First of all is the wavelength, the frequency of compression and rarefaction waves. Higher frequencies will come more often than lower frequencies. And the second thing that we have is amplitude, the intensity therein. Both are encoded by population coding, but there may also be a frequency coding and action potentials as well. And we're going to allude to that a little bit in the mechanism when I show you the mechanism that's occurring. You don't have to memorize all of these steps. These are not steps that I'm going to ask you to memorize. But I do want you to understand what's going on from a physiologic and pathophysiologic perspective. We have air waves, compressions, and rarefactions that are getting turned into mechanical energy. They're being transduced on the eardrum or tympanic membrane. And you have these three amazing bones, the malleus, the incus, and the stapes, which is named after a stirrup on a saddle. That mechanical energy then is transduced into fluid waves inside the organ of Corti, and then we'll have hair cells in a little bit. But there are times when you have hearing loss, and the hearing loss may depend on where it's occurring. For instance, if you have a big old plug of wax, or you get a cotton ball stuck in your ear, or perhaps a malformation, that sort of a thing, you can have a hearing loss that originates from out here. You can have a hearing loss where there's fluid buildup in here. We call that otitis media. Or in some cases, you can actually get deposits in here and so forth, or loss of bone structure. And then you can have issues with the hair cells. You can have issues with the nerve, and you can have issues with the higher order neurons in the brain. But from about here at 6 out, we would call this a conductive hearing loss. And these guys here, the nerve and the organ of Corti, we would call that a sensory neural hearing loss. And then if it was up in the brain, we'd call that a central hearing loss. Actual sensory cells are contained in what's called the organ of Corti, which contains several tissues. That's why it's an organ. And it's a double duct kind of a situation. It's got this double chamber of fluid. And inside, and normally it's rolled up into this snail-like structure. The cochlea is the Latin word for snail or screw. But you can see in cross-section here and here, that's where the real action is with hair cells that contain these structures called stereocilia. And I'm going to break out this mechanism for you. So we have these fluid chambers, and there's actually a second fluid chamber. And here are the hair cells down here in purple. The hair cells are embedded in a one membrane, and then the hair cells are juxtaposed to this other tectorial membrane. The one membrane is called the basilar membrane. This right here, these blue cells comprise the basilar membrane, and the pink here, that's the tectorial membrane. And the hair cells are juxtaposed between the basilar and the tectorial membrane. Let's go down to the level of the hair cell for a moment, and then we'll talk about discrimination of pitch and intensity here in a second. So each hair cell has 2 or 3 sets of stereocilia, these specialized projections, and they're actually linkages, protein linkages in between them, so they usually don't behave independently. In any case, when there is no sound, when we have quote unquote the sound of silence, these hair cell channels, these are mechanoreceptor channels for sodium, are partially open. So the sound of silence is actually a basal tone of action potentials, a basal frequency of action potentials. And so they're firing off with some regularity. There's a little bit of neurotransmitter there, so we're getting action potentials, firing off, maybe at regular intervals. And then when we have sound, you have wiggling of these stereocilia, and so you'll have periods of compression. Again, that's why relative refractory period is so incredibly critical. Why I'm belaboring it a little bit. When we have sound, we're going to vibrate one way and we're going to get a compression, and then we're going to vibrate another way, and the channels are going to close when they get deflected the other way, and we'll get a rarefaction. So we get this alternating high frequency, middle, low, high, middle, low, high, middle, low. Understanding this phenomenon allowed us to develop cochlear implants. And if you want a good ugly cry, look at somebody who has had their hearing restored with a cochlear implant, or a mother who sees her child hearing for the very first time. It will knock your socks off. And it was that decoding of the frequency coding of the stereocilia that led to cochlear implants. But there's more to the story, and there's always will be. But again, when we're not stimulated, we have a basal tone of frequency, and then when we are stimulated, we get alternating dense frequencies and rarefaction frequencies. We get to population coding when it comes to these stereocilia. Well, that's where the basilar membrane is believed to come in. The hair cells sit between the basilar and the tectorial membrane, kind of like they were sitting between two arms of a pitch, of a tuning fork. But it's really, the data seems to indicate that it's the basilar membrane that really drives the resonant frequency of vibration. What do I mean by that is that it varies in thickness. So at the proximal end, where the sound waves sort of enter the fluid of the cochlea, the membrane is relatively thick, and it will vibrate at a certain pitch. But as we get further and further away, that basilar membrane gets thinner and thinner. And the thinner it gets, the lower its resonating frequency is. So if you get a given pitch, it'll only vibrate one part of the basilar membrane, or one region of the basilar membrane. And it'll only stimulate those cells in that region. Now, if it's louder, it'll stimulate more cells in that region with the center on a given wavelength. So that's where we get population coding in the organ of Corti, is with the basilar membrane vibrating, which then pushes the hair cells and the stereocilia into the tectorial membrane. That's also one of the reasons why percussive sounds like drums or gunshots are incredibly damaging to stereocilia, because what happens is the intensity of those sounds, especially at close range, is so intense that it'll literally ram the stereocilia into the tectorial membrane, damaging the stereocilia to where they're stuck either permanently open or permanently closed. And we detect that as a frequency change, and we detect that as pitch. That's why one of the things you should take care of is your ears. If you're going to go to a place that's loud, you should wear protection. You should take care of your ears, you should take care of your skin, and you should take care of your eyes. Doesn't mean you can't get tan, but there is certainly an overdose. Doesn't mean you can't go enjoy live music and be involved in that, but protect your ears, protect your eyes out in the sun. Those things will help improve your quality of life over your lifespan. But it's that vibration of the basilar membrane that pushes the stereocilia into the tectorial membrane. It looks like the tectorial membrane is being wiggled here, and it probably wiggles a little bit, but it has a constant thickness. There is no physical basis for differential vibration of the tectorial membrane as we understand it. But we see a very clear change in thickness of the basilar membrane. And so there, I just have the arrow there going to the basilar membrane, showing you where it is in the cross section. But that combination of frequency and population coding allows us to discriminate not the pitch. So we have population coding, okay, different pitch, population coding, different pitch, population coding, and also intensity, but also that compression rarefaction pattern that we see will also help us discriminate pitch. And I've already mentioned this, but here's a figure for you. Conductive hearing loss is basically anything that involves the middle ear out. So the outer, the pinna or the structures of the outer ear, the ear canal, the tympanic membrane, or the three bones. That's called a conductive hearing loss. Interestingly enough, if you do certain tests, you can actually discriminate between a conductive hearing loss and either a central or a sensory neural hearing loss. Why? Because with certain tests where you're using bone conduction, like with the skull, conductive loss, you will hear the sound louder in the affected ear than in the non-affected ear. Central and hearing loss, you do not see that difference. You see a deficit in the affected ear, whether it's the organ of Corti or the cerebral cortex or the nerve that serves the organ of Corti. Balance is not far departed from auditory. It uses stereocilia, but in a slightly different way. Like in the organ of Corti, the vestibular side of the vestibular cochlear apparatus includes a fluid-filled chamber with hairs that have stereocilia. These hair cells are in these supporting cells, and then they have sort of this gelatinous material that sort of surrounds them. And what happens is that the fluid in here moves, and that causes deflection of the hair cells. The book uses this brush moving against a stationary board. If the brush moves right, the hair cells deflect left, and so forth. And that's what's going on. Have you ever been walking around with a drink that has ice cubes in it? And if you notice that if you turn to your left or turn to your right, the ice cubes kind of stay in the same place. They kind of rotate in your hand. Well, that's what's happening in the fluid. The fluid has momentum, and as you turn, the fluid resists that change in direction, and so it will deflect the hair cells as you turn your head. When you turn the other way, the fluid deflects the other way. If you get into a chair and spin around for a while and you stop and you're dizzy, that's because the fluid is still circulating in the semicircular canals even though your head is stopped. And that's causing a phenomenon known as nystagmus. On the other hand, there's another part of the vestibular apparatus, the vestibular cochlear apparatus, called the macula. This is about head position. And here again, we have fluid, and you can't see it here, it's not shown. You have this gelatinous material, but now we have this additional material that are literally organic crystals. They're called otoliths. They are calcium, phosphorus type crystals that are heavier than the fluid around them, and they will settle in gravity. This is a gravitropic situation. Yes, it's mechanotropic, but it's gravity that's driving the deflection of these hair cells. So a gravitropic phenomenon. And so when the crystals settle on the hair cells, it deflects them. Under normal circumstances, population coding will drive this. Which hair cells are being deflected tells the brain what the position of the head is. So when we're upright and our crystals are in the right place, they will deflect a certain population, causing them to depolarize more frequently. If we tilt the head, we'll have changes in deflection patterns, changes in frequency coding, changes in population coding, leading to a perception of a head tilt. And nystagmus and the vertigo associated with nystagmus are one of the most common disorders that we can encounter in everyday life. It can be transient. For instance, you can get out of bed and have episodes, or it can be somewhat persistent. There are certain types of maneuvers that they use in order to redistribute the crystals to help alleviate issues of nystagmus. Nystagmus is basically a reflex where the eye muscles are trying to compensate for head movement. Your eyes gimbal inside your head, which is basically an organic way of image stabilization, which is why it's so hard to do video. It's easy to get seasick watching somebody use a video because the video camera isn't gimbaling like your eyes do, and that's why video looks different than your eyes unless it's stabilized. And so when the eyes are trying to compensate for inappropriate head movement, you get this sort of phenomena. It's called nystagmus. And we are going to stop right there. We're exactly at 30 minutes. So thank you for your time, and we will resume with vision in part D. Vision is our poster child special sense because of all the things it has the most profound impact on quality of life if for some reason we depart from normal vision, if you know what I mean. So thanks for your time, and I will talk to you again soon.